Welcome to Teach Yourself About Sleep. This video is the fourth in a series of videos on the topic of sleep. REM sleep. Sleep consists of two main phases, non-REM and REM sleep. In non-REM sleep, body functions are silenced and muscles relaxed. Sleep begins with non-REM phases, and particularly the deep non-REM phases appear in the beginning of the night. In REM sleep, many body functions become irregular, like the body would be detached from normal regulation by the autonomic nervous system. During REM sleep, muscle tone almost disappears, leaving the sleeper without ability to move. REM sleep appears towards the end of the night. About 20% of human sleep time is spent in REM sleep. REM sleep has two names. One, because during certain phases of REM sleep, eyeballs move under closed lids, giving rise to the term rapid eye movement. The other name, paradoxical sleep, refers to cortical activation in the middle of sleep, which is a paradox. A prominent theta rhythm is a sign of REM sleep, particularly in rodents. REM sleep can be further divided into phasic and tonic sequences. Eye movements and muscle twitches define the phasic REM sleep. One of the most prominent features of REM sleep is the disappearance of muscle tone. Yet another mysterious feature is the unexplained activation-deactivation of autonomic nervous system, leading to bursts of heart rate and blood pressure rises, peaks in breathing, and loss of proper temperature regulation. Large part, but not all, dreaming takes place during REM sleep. Here are some examples of recordings during REM sleep. Eye movements can be observed from the EOG recording. They coincide with the PGO waves that can be recorded from the lateral geniculate nucleus. Here is an intrahippocampal recording showing the prominent theta activity during REM sleep. Basal cortical activity is maintained by secretion of several neurotransmitters into the cortex. Neuronal groups from brainstem, pons, hypothalamus, and basal forebrain send their projections to cortex, and when active, release their neurotransmitters to activate cortical neurons. Neurotransmitter release in the cortex has specific features in REM sleep. During waking, neurotransmitters from all neurons, including serotonergic, noradrenergic, histaminergic, and cholinergic, are released actively. During non-REM sleep, release of all neurotransmitters decreases. During REM sleep, all other neurotransmitters almost stop their release, but acetylcholine secretion is increased to the level that it was in waking. This means that the high cortical activity that we observe in EEG during REM sleep is supported by only one neurotransmitter, acetylcholine, while during waking, several other neurotransmitters contribute. Regulation of REM sleep appears to be a very complicated combination of activities of different neuronal circuits regulating both brain and muscle activity. New methods of neuroscience have revealed more neural circuits that contribute to this regulation, and most probably more will be revealed in near future. Here is a summary of what is presently known about the regulation. In the drawing, Nuclei that are active during REM are marked with red, and those that are inactive are marked with green. Sublaterodorsal nucleus, SLD, is in the heart of the regulation. It is inhibited by noradrenergic locus coeruleus and serotonergic raphe neurons, which also inhibit the cholinergic LDT-PPT neurons. GABAergic VL-page also inhibits SLD. When REM is initiated, LDT-PPT activates SLD, which inhibits VL-page neurons, releasing its own inhibition. SLD activates nuclei in the ventromedial medulla, which further inhibit VL-page. LDT-PPT activates also the cholinergic neurons in the basal forebrain, resulting in hippocampal theta. There are two theories of the events resulting to muscle atonia. According to one, the ventromedial medulla nuclei inhibit spinal motor neurons. According to the other, these neurons are inhibited through premotor neurons activated by SLD. REM sleep has sometimes been called also dream sleep, but it should be noted that dreaming takes place also during non-REM sleep. One of the consequences of decreased muscle tone during REM sleep is that we cannot act out our dreams. Unsuccessful coordination of components of REM sleep results in dramatic disorders. If during REM sleep muscle atonia is lacking, the dreamer acts out his or her dreams, often very violently. On the other hand, if REM sleep is initiated directly from waking, the resulting muscle atonia induces a collapse. This is what happens in narcolepsy. As you notice, it is important to get your REM sleep right. Sleep well.